المؤتمر الدولي للجمعية المصرية الألمانية لعلم الحيوان. نرحب بحضراتكم جميعا. والحقيقة في البداية بحب أشكر الأستاذ الدكتور بديع الأعرج والأستاذ الدكتور فتحي عبد العاطي على هذا المؤتمر المتميز، وأيضا اللجنة المعاونة. والحقيقة امبارح شفنا يوم رائع حضر فيه الأستاذ الدكتور عصام شرف والشيخ ناجي الجندي، وساعدنا جميعا بهذين المحاضرتين، وايضا ساعدنا بالكلمه الافتتاحيه التي قالها الاستاذ الدكتور فتحي عبد العاطي. الحقيقه برضه هذا المؤتمر تميز حسب برنامج الموضوع بالستيودنت سيشن، اللي هو الجلسه بتاعت الخاصه بطلاب الدراسات العليا، وهذه نقله جميله واستجاب لها الاستاذ الدكتور جابر البشار رئيس جامعه القاهره، ورصد 10,000 جنيه للمتميزين في هذه الموضوعات المقدمه من هؤلاء الشباب. ننتقل الان الى جلسه اليوم. سنستمع الى الاستاذ الدكتور ايهاب الضبعه، بروفيسور and head of biochemistry and molecular biology department في يورب هاريس ريسيرش انستيتيوت. وعنوان المحاضره Vaccination in Infectious Diseases. وان شاء الله نستفيد بهذه المحاضره ونشكر حضرته مقدما وليتفضل سيادته. والحقيقه انا بشكر وبسعد ايضا بوجود هنا في مصر الدكتور شهيد زميلنا من نص قرن مضى، فسعداء بهذا اللقاء. ويتفضل الاستاذ الدكتور ايهاب.
السلام عليكم. مناسبة سعيدة ان احنا نشوف الجمع العلمي الممتاز ده.
ان شاء الله بس هتبقى حضرتك يعني خلي بالنا من المكتب علشان برضه نعمل وقت مناقشه 10 دقائق
ان شاء الله.
بس انت اتفضل ان شاء الله.
ان شاء الله.
اتفضل.
باذن الله. زي ما حضرتك قال انا بطلب رئيس قسم الكيمياء الحيويه والهندسه الرئيسيه بالمعهد الدكتور ايهاب. شغلنا بيتحسب في ديفلوبمنت فاكسينز، وحاليا R&D development for recombinant therapeutic protein للإندستري. Today we'll discuss one of the top issues, vaccination and new trend in vaccination. As we know, vaccination is a vital immune system with infectious diseases of the small body to induce infectious or invade to induce protection or combat for future infection. Since Edward Jenner has discovered this vaccine, and all of us know about vaccines, and there is big benefits for the human kind. There are conventional vaccines, the traditional ways, life-innovative vaccines, inactivated organisms, subunit vaccines, and recombinant vaccine and toxoids. Also, this type of vaccines help in many of diseases, but still have problems. For example, the return of virulence in life-limited vaccines that we have checked in polio vaccines, and in poor immunity or non-native structure of the protein for the subunit vaccine and the recombinant vaccine. And one big problem is that the uptake of those vaccines is uptake by immune system as exogenous protein, and so presentation is MHC class 2, which is not efficient. Okay? It's only induced antibodies, and so why there are need for a new methods, because many of diseases have no solutions or no vaccine till now, and it's, as you see, many of people all over the world. TB, HIV, diarrheal disease, and many other viral and bacterial infections. So simply, what's DNA vaccination? In 90s, few scientists noticed that if we take a gene of a pathogen, for example, here is the virus, if we take one of its genes, clone it in vector capable of expression in mammalian system, and injected intramuscularly in mice, they found that the gene expressed first is plasmid, transfected into the nucleus, and RNA is produced, and the protein is expressed. And also the muscles does not have the MHC class 2 and the class 1. They found that both arms of the immune system, cellular and humoral immune systems, have been involved, which was so strange. DNA vaccination have a very characteristic immune response and own characters, which is completely different from the routine vaccinations ways we know. For example, the vector itself, the backbone of the vector, the plasmid vector, it's a DNA backbone. It's contained what is called CDG motifs. It's a consequence of DNA and nucleotides. This motif, through the toll-like receptor, induces the proliferation of B cells, dendritic cells, macrophage, and natural killer cells. It's innate immunity. It's related to the antigen. Also, it's induced interferon alpha from 12 and interleukin 18 to produce interferon gamma, which make Th1 polarization. Before, we cannot make the Th1 or Th2 polarization. It's very important, especially in autoimmune diseases and some infectious diseases like parasite disease. Also, it's a double strand through the sting and the TBK1 receptor make induction of innate immunity, general immunity. That's only in the vector, relative to the antigen. If we look to the antigen itself, what happened exactly is that when the DNA vaccine enters to the cell, it's entered to the nucleus and the RNA is expressed. It's expressed and the protein produced as intracellular. Exactly, it's like the native viral infection. So why DNA vaccination is a very potent. You cannot do this by traditional way. And so it's like a viral infection occurring, natural viral infection occurring, and it's made endogenous. MHC class 1 is used and induce cytotoxic T lymphocytes and a very interesting thing for DNA vaccination. And so, and also this protein is excreted outside. This protein either produced in endogenous this way and induce humoral cell and cytotoxic T lymphocytes and T cell memory, or this protein is secreted outside these cells and taken by other immune system cells as exogenous one and do the traditional way. And so, how it evoke both cellular and humoral immunity, especially cytotoxic T lymphocytes. And also there is a very important thing, it's cross-priming dendritic cells. That's the DNA constant. Either introduced directly intracellular and so induce the exogenous way, or induced directly or induced into dendritic cell, or what called is cross-priming. With the cross-priming, the macrophages and other cells undergo apoptosis and eaten by dendritic cells and expressed also endogenous way. So all of these pathways cannot find in the traditional vaccine. So it's very powerful tool. The vector itself, we clone the gene. Before the gene, we put the Cossack sequence because it's a mammalian expression system. And we take the gene from virus, from whatever the organism has in the gene. The leader sequence because it's make good expression, and poly A to make a good translation of the protein, and controlling element for replication of this vector into the bacterial cell. There is now FDA-approved vectors for trials in human and high primates. The application of DNA vaccine is autoimmune diseases, especially its property to control the Th1 Th2 polarization. In cancer, you can augment to specific genes, to specific cancer, and in infectious disease, but that's beyond the scope of this lecture. But it's magnificent working is done right now. How to design the DNA vaccinations? First, you select the gene. For example, in human papillomavirus, the L1 protein, you find in the L1 protein the E6 and E7. Those two proteins, if you express it intracellular, it's automatically polymerized together and give you empty virus. You produce empty virus. There is no genetic material, and so it's not infectious, but it's a virus. First, you have to select the proper protein, and you have to work a lot in this, because it's serious. What type of immunity you think you want to issue? It's a big issue. Second, you make a construction of the DNA vaccine. Take the open reading frame, put leader, cossack sequences, and ribosomal binding sequences, and the poly A, and put it in proper vector. And then you have to make a demonstration. The protein is going on working or not. This demonstration through, we may put the protein fused to fluorescence, fluorescence protein, like this issue, and so you can know if it is expressed or not. You can measure by Western blotting. You can put SCP40 origin of replication, and so you can express this protein in host cells, in tissue culture, and so you can detect if it is working or not. Also simply, you can inject it in a muscle mice and know it's working, it's producing the protein or not. That is the first step. Second, you can augment protein expression, as we said before, by cossack and upstream region in the DNA or in this second structure. You know, the early sequences control the binding. That's a payload, that's natural for ribosomal binding site in the eukaryote. But if you have second structure making another payload, you make a problem. And so you have to look good in the 5-9 coding region and ribosomal binding site. Second, not every codon is used by every organism. For example, bacteria and viruses have a codon for amino acids. It's rarely used by human. For example, actually have 5 codon, but only 2 or 3 used by human. But the other codon used by viruses and E. coli cannot be expressed. And so this protein will not translate. There is hang-on translation. So you have to make good codon usage. Make synthetic gene or change these codons to make the gene expressed well in human or in the host, whatever is the host. You adjust the codon usage to the host. Then you can increase the cells in producing this protein through transfiction agent, like transfiction. Many other brand names is served right now. Or you can use viruses or virus proteins which invade the cells and make more copies of your DNA and more copies of your protein, so you increase the expressed protein to increase the immunity. Second, you can augment immunogenicity. How? As I said before, it's a molecular biology tool. So you can put a lot of things in it. You can direct to specific epitope. You can so simply put in your protein B or T cell. You can use immunomodulatory and immuno-enhancing molecules and genetic adjuvant like cytokines. You can make your protein fuse to cytokine or make co-expression of cytokine. These cytokines manipulate the system and direct the immunity towards the way you like. There is something very good. They make prime boost strategies. They discover that if you make repeated vaccination with the DNA vaccine itself, it's give you limited immunity, and that is a seroconversion. But if you do it protein and DNA, it's give you slightly higher than this. But very strange, if you make DNA vaccination, then prime by the protein, you got this. Very strong immune response. They found it happens when you prime by DNA and make post either by the protein or viral vector expressing the protein. And as you see, it's make a big difference. Second, you can make direct or intracellular routing of your protein. For example, you can go to your protein leader sequence which direct your protein endoplasmic reticulum, and so it will be through MHC class 2 and give you antibody, or towards cytosol and enhance the CD8 instead of CD4 and give you cytotoxic lymphocyte. DNA delivery and administration rules. It's make big difference. Every route has its characteristics, its ways, its immune system you can induce. For example, you can simply inject hypodermic or intramuscular, but in this case you need big amount. For the one max you need from 10 microgram to 1 milligram. You can use other technique. You can put your DNA on a golden particles and convert it using the gene directly to the skin. And so you need very small amount, 16 nanogram to 20 micrograms. But this induced pathway, and this is induced another pathway, mainly the dendritic cells of the skin. You can use aerosol or cationic liposomes. Mix just your DNA with cationic liposomes and make aerosol nasal, and so you can induce mucosal immunity. So simple and it's very efficient, especially in animals and cows and farm animals. You can use intracellular bacteria like Shigella or Listeria. Just put your vector into the intracellular bacteria and raise the bacteria of modified strain, invade the cell, and so it will be a good vehicle to transfer your DNA intracellular, and it evokes the immune system in a completely different way. You can use nanoparticles. Put your DNA into nanoparticles, whatever is nanoparticle, or magnetic nanoparticles. And so you can bombard directly the dendritic cells or inject directly into any tissue and give completely different profile of immunity. And also the route of administration is intramuscular, intradermal, intraperitoneal. Intraperitoneal does not work so good in many traditional ways, but it works very good in DNA vaccination. We show this in our experiments. And also, you can do it oral, just by spraying the mouse, which is very simple, not invasive. Examples for the DNA vaccination in infectious diseases. The viral infection is the perfect for the DNA vaccination. Why? Because we said before, it's like intercellular expression. It's like natural viral infection. It's mimicking exactly the natural viral infection, but without hazards or pathogen. And it's induced cytotoxic lymphocytes and B-cell groups very efficiently. And that is #1 candidate for the infectious diseases. There is infectious disease model. From the early models, the work was the influenza virus. You can see this orange line and this green one. The green line is the survival of mice after days of infection with influenza. And that's DNA vaccinated mice, vaccinated by nucleoprotein, intramuscular, just 2 shots. It's still survive. Okay, maybe not eradicated completely the virus from the blood, but still survive. HIV, brain, Ebola, rotavirus, all of this, there is a successful experiments going on. And in the clinical trials, now, HIV, influenza, human papillomavirus, and HPV. In human papillomavirus, as you know, it's infect the vaginal system of women, okay? They could express the T7 and T6 protein and make empty viruses, and it works well till now, and it's near to be approved. In bacterial infection, as we said before, we will encounter expression errors because viruses usually expressed in humans or in the host, but bacteria does not express their proteins in the host system. And so we should go through the expression errors for the usage. There is carbohydrates in bacteria. You cannot express in human, but you can express the proteins which conduct these carbohydrates. And you can go through all of this by fish display and anti-antibiotic antibodies. The antibiotic antibodies is a mirror image of the antigen, so you can make antibiotic antibodies for carbohydrate proteins using this technique. There is efficient disease models working, the research is working very efficiently, like the Mycobacterium tuberculosis, okay? It's now rising up again, and in Europe it's a big challenge. Also the Helicobacter pylori, which infects the stomach and found in a lot of people without saying this. They use the USP vaccine and OPA protein and with adjuvant and interleukin 12. And in the DB, they make second priming by the BCG. And it's very good, gives them very good results. And now mycobacterium tuberculosis about passing the clinical trials. The parasitic disease is the most complex because you have a complex life cycle. You have multi-structures, surfaces, with changing surfaces. And so the challenge is big. And short improper presentation because the parasites stay open, fertile in the blood without anything from the human. And so, sure, they have improper presentation for the system. And also, they develop their systems for the evasion of immune system. So that is a big challenge. The prime host adjuvant, you can use multivariant. You can use more than 5 or 6 genes, 3 to 6 genes, and inject it simultaneously. And they can work together to combat this. There is official disease models in Schistosomal National. SNV20 is now approved, used in approved vector VR 1020, and it was 47% protection. We got much better than this in our labs. For malaria, it gives 100% survival. For Plasmodium ureae, using nanoparticle coded plasma, encoding MSCP1C terminus protein, done intraperitoneal and intravenous. And now, malaria cocktail vaccine, which include antigen and cytokine adjuvants, now is in last stages in clinical trials. And in conclusion, what is the advantage of antipotentiality? It has a big advantage and potentiality. So simply, you express the native structure of the protein, okay? Especially those for viruses, which is the original host. The exhibition system occurs in the original host. You can generate cytotoxic lymphocytes, antibodies, and T helper cell. And you can control the T helper cell using the tools. You can switch from helper 1 to helper 2. You can direct it, whatever you like. Have a magnificent tools. And no virus or adventitious agent. And stable and safe. It's stable because you transfer lyophilized DNA. No problem for lyophilized DNA. It can stay for months in room temperature. And it's safe, no problem in it. And it's specific. For example, some proteins have common motifs between the protein and human, and so it can develop autoimmune. In DNA vaccine, you can simply remove these parts of the gene, and so you can express the peptides of concern, the peptides only unique for the parasite or the infectious agent. It's cost effective. What you do, that is industrial scale production for the DNA vaccine. You have a fermenter. After fermentation, you break your DNA, you make primary purification and column purification, and then you get it. It's a generic platform. You can do it for any vaccine, no specification. So very simple in industry. And also, it's faster vaccine development than any traditional method. For example, in epidemics, for influenza, flu epidemics in 1980, there were more than 20 million deaths. Why? When they discover a new strain of virus, rise, it takes at least 6 months to isolate this new strain, to culture this strain, to identify or make the attenuation research, and then at least 6 months to release a new vaccine. In DNA vaccination, it's completely different. If you are using some gene in the old strain, simply use conservative primer and modify the same gene, whatever is the changes or the mutation in it. And so simply clone it in a bacterial plasmid, make culture, and release it. It takes maximum 1 month. Very simple way. And that's very powerful too. And also, it's very strong vaccine discovery platform. In usual way, you have to separate single protein by protein, or express protein by protein, and it will not be in the native form. But simply, for example, if it is a virus genome, you can chop this genome into pieces, clone it in expression vector, and check every isolated plasmid in a group of mice, and so it's a very fast platform for recognition of infected antigens or genes. Intradermal Health Research Institutes, one of our aims was to develop vaccine to Schistosomal monsoonii. Using the molecular biology techniques, we develop libraries. We have different libraries, expression libraries for different life cycle stages, adult worm, Schistosomal, and Circaria. In London, that's the expression libraries. And this library is distributed all over the world. You can find it in the gene bank and many other mirror images all over the world. We select genes from library screening using immune-specific methods like irradiated Circaria antibodies, methods which prove the antibodies prove to be successful in protection, like irradiated Circaria mice and other methods. We isolate candidate genes. We isolate 7 candidate genes. Genes with product in BCDE1, that's expression vector in mammalian cell, and check this for protection against Infectious disease in mice. What they found, that's some of our publications with this. For example, when we check tegmental antigen, SM21 gene, it gives us 41 in Swiss albino mice. But very strange, when we change the strain of mice, it gives us 53, which shows a muscle, glycolytic protein, metabolic protein, but it gives us no protection. Also, it reduces the granuloma cells. And when we choose muscle protein, it's away from the surface. That's a surface one. That's away from the surface. It gives us higher protection. Intramuscular. All of this was intramuscular with Swiss albino mice. It's higher protection. Also, it is not exposed to the surface. So why? We have to dig a lot to find what's your proper system. Second, we make a cocktail vaccine. 2 genes on the same vector, B-BAT vector. Very strange. We got 56, very high. That is the highest level we got for protections. 56, even more than the internationally approved for clinical trials now. Second, we combine protein and DNA. All the literature claims that if you make a post with protein after DNA, it gives you higher. It gives higher, but still, the cocktail vaccine is much higher than this one. As I said before, you have to make a lot of work and you have to discover yourself, especially in parasite, complex parasite system like Schistosomes. When we make DNA vaccination after parasitic contact, we tell ourselves, okay, some people may get treated, they will not get use of the vaccine. We check mice after treatment with parasitic contact. It gives high anti-pathological effect. You look to this, that's only parasitic treated mice. And that's fibrin DNA vaccinated. It's increased the fibrosis. When we made fibrin DNA vaccination after parasitic you can see this. We mostly eradicate the fibrous granuloma. We cannot explain this, but that's the fact. We can use. After this, we check different mode of vaccination. We use the SMR in intramuscular to give no vaccination, but in subcutaneous and reduce the dendritic cell supplementation to give us higher fragmentation, but still moderate. We got very great protection when we do it intraperitoneal, and it gives us massive reduction in over and worm. It's also make a conflict with the egg-laying system of the parasite. And finally, what is the recommendation? That's all good news, and that's all good techniques. What we should do? We are a developing country, and we should work what is near to the market. We skip a lot of our research now, including the Schistosomiasis research, because it's an academic research. We want something to apply. And so, what's my advice for those who are work with DNA vaccination? As we know, vaccine is a cost-effective industry. India is the #1 producer of vaccines in the world, and they are the developing country. They were like us, but now we are bad. DNA vaccine is simple and cheap, and does not need big skills. And its production is integrated process. From research to industry, it's a few steps. That's the chain, concomitant chain for production. You make a large scale, you make pilot scale, and then you make the acceptance and validations and put it to the market. In DNA vaccination, it's short chain. It's not like developing a vaccine from protein or virus. And human vaccines have a lot of ethical problems, biosafety, and approvals from different committees. And so, my big advice is that to look to veterinary DNA vaccination. Concerning animals, the biosafety issues, the ethical issues, and the approval issues is very simple. Second, in Egypt, more than 90% of our animal vaccines are imported. Also, it's the viral agents. So it's easy to develop a DNA vaccine for viral agents. And so, it's a big market in Egypt. Nobody knows how to. It's a very big market in Egypt. For example, for influenza, H1N1 vaccine for flu, it's millions, not millions of doses per year in Egypt. And so, it's a very good market. It's simple research. You don't have to go through ethical biosafety and approvals as you do for a human. And we have to build up capacities. We have in our lab, for example, limited pilot scale production. We make R&D in the lab and make pilot scale production through fermentation, purifications, and other stuff. And I'm sure there are many units like this. It's easy to use such units and such facilities to develop vaccines for veterinary. And finally, thank you, and I hope I'm not annoying by now.
Thank you, Dr. Eihab, for your marvelous and interesting lecture. And now, any questions, please?
شكرا دكتور إيهاب للعرض الجميل للفكرة الأساسية. الكويشن بتاعي about you mentioned cocktail vaccine twice when you are talking about malaria and when you are talking about schistosomiasis, schistosomiasis. First question, is it, if we said cocktail vaccine, is it specific or not? And the second, you mentioned in malaria, now they are at the end of clinical trials. Please give me more information about this point. Thank you.
Sure. When you start with a DNA vaccine, you choose a specific gene. It's not a specific gene for the organism. No. It's specific concerning the immune response. For example, there is surface tegmental antigens. You found all those antibodies for these antigens, okay? But not cellular response. So simple, it's exposed to the surface, and the parasite, all over thousands of and millions of years, develop his own system. He manipulates our immune system. He gives us the antigens which is not working for him. Okay? And so you have to select carefully your protein. It's not only specific for the parasite. Sure, it's specific for the parasite. But you should imagine. For example, when you use the surface protein SM21, we didn't have this big issue. When we use intramuscular protein, it is not exposed to the surface. Why it should work? It's hidden. The parasite hidden its important proteins and expose non-important proteins. And so you should have a very smart approach depending on your experience in the immunology of this parasite. It's a big issue to decide which antigen. As we know, a lot of parasites now working is going to the recombinant virus vaccines and DNA vaccines. Until now, little vaccines developed. Why? Because it's controversial. I worked more than 25 years for development of vaccine for mice. And finally, I decided to quit. And now I'm working in developing recombinant therapeutic proteins. We produce now interferon alpha, growth hormone, because it's very straightforward. I just got the gene, and that's what I meant by near to the market research. Not as in the United States, in Brazil, we were a member of huge genome, just genome projects. The genome project finished, but they didn't find a vaccine. And now, big institutions on the world that spend millions of dollars, but they still not able to find the vaccine. It's a big issue. That's your first question. Your second question about malaria, as we said before, there is a protein, MB123. It's not my field, okay, but this protein and another protein, they claim a tail protein vaccine works well in the research. And they claim, and I said a lot of lines under claims, you don't have to agree with everything you hear. Okay? For example, just mice. Capron claim, and they claim that with glutathione transferase, he make a magnificent work when they go to repeat this in the field with baboons, with mice, even with mice. It was experiment with mice. They found nothing. And so, the type of mice, the immune system of the mice, and mice is completely different from human. And so, you skip everything to the garbage. It's a completely different immune body.
Thank you.
Yeah.
شكرا جزيلا طبعا على العرض الجميل والمحاضره الهائله. هو في بس استفسار يعني حضرتك جرت العاده ان الناس بتقول ملاريا ويقولوا مش السيزونيازيس، بينما انا اعتقد معلش ممكن نصحح مع بعض هو المفروض البارازايت يعني الملاريا دي ريزلت ده ديزيز يعني I cannot vaccine myself against disease يعني disease دي نتيجة لأنه يبقى فيه طفيل معلش ده يعني حتى عالميا موجود هذا الاستعمال اللي هو غير دقيق يعني حضرتك شيستوزوما مختلفة جدا عن الشيستوزوميازيس والبلازموديوم مختلف جدا عن الملاريا فمش عارف رأي سيادتك ايه؟
لا it's scientifically very broad بس you develop vaccine for the parasite and also you develop vaccine for the disease.
ما احنا حضرتك معلش خلينا نتكلم الاول في فروم ساينس
يعني لا بيقولوا الفلو فاكسين يعني
انا بدرس
ممكن يقول انفلونزا فاكسين you can say the flu vaccine
يعني انا بتكلم على البارازايتس اللي هي بتبتدي من الايوكاريوتيك سيل في البروتوزوا لغايه لما نوصل للفريجنسي في الهيومنز بيعتبروها حتى بارازيتيزم يعني فالمفروض ان انا اللي بيعمل الكوز هو البارازايت فانا بقول الافكت اوف بارازايت ما اقدرش اقول ابدا الافكت اوف شيستوزوميازيس ولا اقدر اقول
لا افكت افكت بارازايت بس انا بتكلم على الفاكسين could be developed I can say literally with a parasite or vaccine with a disease because I'm speaking about vaccine I'm not speaking about effects I'm speaking about vaccine and all vaccine to the disease itself or to the agent infectious agent and most of them.
شكرا.
Thank you very much for this.
You are
very wonderful presentation. My question is, how long this type of DNA vaccination stands in the body? Is it long term or short term? I mean, have you followed the immune system in the animals?
Sure, it's short term. I mean, every protein you express decay. Sure, it's short term, okay? Because the protein in the body, there is proteolytic enzymes, scavenging enzymes. It's get rid of the protein, okay? And so why? But the immune system you are evoking, especially evoke the memory cells, is lost. But the protein itself, no. It takes maybe a few days and sometimes it takes months. And hardly to digest the proteins, it takes months. And sometimes they don't know how. If you put it in a viral vector, could keep. But the problem in viral vector, there is immune response to the virus, to the carrying virus, and so cause inflammations. And sometimes, if you keep the protein expressing, you get the opposite. It means you get non-response, agony, which is silencing the immune response by exposing the body to this protein. And so it is not good to make it lasting exposure. No, it's good to make pulse exposure.
The second question is, can we use the same approach for gene therapy?
It is a gene therapy approach. We take this approach
because it's very simple. Not use
from the gene therapy.
Yeah.
In the gene therapy, they express the protein to make it express intracellular to fix something.
Yes.
We do the same, but our purpose to induce the immune system by this protein, not to fix something.
I mean, I'm speaking just about time-wise. How long this immune response will be our
For example, if you take a measles vaccine, it's keep for all of your life. It depends on the pathogen, a lot of things. Usual vaccination now, it's keep for life.
For lifelong. Thank you very much.
Yeah. Any other question?
Ebola virus?
They are trying. You know, Ebola virus was a limited number of patients. But now there is a breakthrough.
Limited number?
And now there is a Canadian labs claims that they got a DNA vaccine for Ebola virus. And I said claims. Okay?
But there is no clinical trial?
No, not. I think not. I tell you something. I don't know everything about every single day vaccination in the world. I do a small number of research, and I know some bits of information from Zealand. But in details, I cannot say what is it.
يعني نسعد اسم كل صاحب سؤال عشان في نوع من الكوميونيكيشن بينهم بعض. هو بس سؤال حضرتك: Is there a specific sequence of such DNA vaccines for each of schizomatopic and schizomatopic? The DNA sequence of the vaccine.
This question I've seen it for 25 years. I tell you something. First, is there a protein in DNA vaccine? You get a candidate protein. You know it works good to induce the immunity you want. Okay? The question is there a candidate protein which can be a vaccine for schistosomes? Sure, there is no one. You can find in the literatures hundreds of the genes and the proteins and thousands of trials. But till now, the vaccine, which gives at least more than 90% protection, is not exist. And if I know the reply for these questions, I will do so.
Thanks. أشكرك على العرض الجميل وفترة قيمة. سأتحدث بالعربية.
طبعا ده أجمل حاجة.
معلش يعني أنا دايما أفضل بعد ما نتكلم بالإنجليزية برضك يعني ملخص بسيط أول. لأن البعض عشان حتى مش متخصص.
تمام. دي برضه معانا طلبة برضه لازم
20% تمام جدا في الموضوع. سؤالي كالآتي ومجرد سؤال استفهامي.
تمام.
يعني لست متخصصا في موضوع هذا ولكن دخلنا على الجين ثيرابي. يعني لو حد بتدخل DNA في جسم صح كده؟
تمام.
ده السؤال الأول. ما مسار DNA هذا مجرد كل مساره في الجسم وصل فين وبيعمل ايه؟
بعد حد DNA ازاي احنا في مصر
مش مهم الداخل
وات ايفر التشعر جزء هيروح انتراسيلر يبتدي يعمل اكسبريشن ويحصل له ديلي بعد فتره.
دي واحده انترسيلر فين؟
في الخلايا في التشو اللي انا لان اساس الدي فاكسين انه يخش انتراسيلر.
دلوقتي انا حاجه كمثلا
يبقى هيخش بلازمتس هتخش جوه المصل لحد النيوكليس.
توصل
لازم ده اساسي عشان يشتغل انها تخش في المصل.
خلايا العضلات فقط.
لا
وليه؟ ما انا عايزك.
انا بقول اول حاجه في التيشو اوف انجكشن هيخش الدي ان ايه ويبتدي يطلع البروتين وهيقعد هاف لايف يبتدي يقل مع الزمن. ده اول حاجه.
معلش عشان
انا هكمل
جوه الخلايا العضليه مش كده؟
تمام.
وصل للنيوكليس جوه النيوكليس.
تمام.
مش كده؟ داخل النيوكليس ما هو في دي ان ايه العادي بتاع الخلايا العضليه.
جميل.
ايه مش هيحصل يعني مفيش اي ازاي هو ده هيبتدي يحصل له اكسبريشن.
عشان كده احنا قلنا ايه؟ في FDA developed vectors for clinical trials. يعني ايه؟ ما هو دي ممكن يحصل كروس كومباينيشن. ببساطه الدي بتاع الفيكتور يخش بتاع الهيومن يخرب لنا الدنيا، يقوم عامل كانسر في المكان الحق.
لا هو سؤال كمان دلوقتي حضرتك كل الكلام ده ابحاث واتعملت آلية تفسير آلية اللحظة ما يدخل الدين يوصل ليه النيوكليوس تمام كده ايه اللي بيحصل عشان يؤدي الفعل بتاعه في الفاكسينيشن ومنصوب بيوصل لكل خلايا الجسد الجسم ولا يعني لانه هنا بقى السيفتي كلها والايثيكال بروبلم اهم 3 نقط.
ده سؤال
دي اهم شيء يعني. وبعدين يعني اتفضل
حضرتك الدي ان يخش جوه النيوكليوس يبتدي يطلع ار ان اي. تمام. كانه دي ان اي من جوه ار ان اي بتاع الجين ده يبتدي يوصل للسيتوبلازم. الرايبوزومز تمسكه تبتدي تعمل له ترانسليشن يطلع البروتين. البروتين ده منتج فين؟ انتراسيلولر.
مش هيأثر على الدي ان العادي بتاع الخليه.
لا
خالص. لا كلمه لا ازاي؟
ما هقول لحضرتك ليه؟ في FDA approved DNA vaccine vector. ايه ده عباره عن ايه؟ شالوا منه كل السيكونسز اللي ليها علاقه بالهومولوجاس والكروس كومباينيشن. يبقى 1. 2 شالوا منه اي حاجه ممكن تنترفير مع البروموتر اكشن في الهيومن. يعني لا في DNA فيكتور FDA approved.
حضرتك الابحاث معظمها على الحيوانات.
لا انا هنا اشتغلت الناس بتشتغل بالاول بالفيكتور العادي لان FDA approved فيكتور ده غالي جدا ومش اي حد يقدر ياخده. لازم تكون حاجه مثلا خلاص قيمته زمالك حاجه يعني بروفد انها خلاص على وشك انك تبتدي بقى الابروفر بتاعته. تمام. احنا بنستخدم دي ان اي فاكسينز عاديه ماميليان اكسبريشنز عاديه اللي ممكن يكون لها تاثير على الجين والجينوم. ان انا اعرف ان الطريقه دي efficient and good ابتدي بقى اشتغل باللي هو FDA approved vaccine. ال FDA approved فيكتور ده فيكتور ما فيهوش اي مشاكل up to our knowledge till now. up to our knowledge till now ال FDA approved فيكتور عاملين FDA فيكتور للانيمالز وبالذات الهايبرامينز كلها وللهيومن. والترايلز اللي شغاله دلوقتي شغاله باستخدام الفيكتور ده وعملوا عليه ريسيرش لا تتخيل في الباي سيفتي والستوكاستيك افكتس والانترفيرنج اكشنز لمده سنين طويله قبل ما توريزي.
حضرتك الجين ثيرابي لا زال فيزيكالي وحتى براكتيكالي مشاكل كثيره جدا.
تمام.
وهو ده ما هو الا لايف من
وما زال
ماده غريبه
ما زال
دي ان غريب بتدخله الجسم بعد فتره ممكن يحصل ايه. فهو الموضوع كبير
جميل. بس الجين ثيرابي بس في الجين ثيرابي هو عاوز دي ان ايه موجود على طول عشان دايما ينتج له البروتين الناقص. هنا حاجتنا كويسه ليه؟ افضل من الجين ثيرابي مور بروميسنج. ما بقاش كل البايوباسيكال بروبلمز دي لان ده حاجه ترانزيانت موجود لفتره صغيره وبيأثر. فانا مش محتاج ترانزيانت بريزنس بتاعه مش محتاج يقعد على طول فبالتالي البايوباسيكال بروبلم بتاعته اقل. وفي حالات مثلا زي ان البقر كله بيموت على الحمى القلعيه مثلا. فيروس الحمى القلعيه لما بيجي بياخد اكثر من 3 4 مليون بقره. البقره تموت ولا ممكن يجي لها حبه كانسر صغننين؟ اكيد حبه كانسر هيتاكل. الكانسر ده ميت طعمها حلو. تمام. فبالتالي في ايشوز. عشان كده قلت ايه؟ الفيترينري فاكسين از ا فيري جود ابلكيشن. ما فيهوش البايوسيكال ايشو اللي الدكتور بيقول عليها، ما فيهوش اكريديشن، ما فيهوش حاجات كتير. وفي نفس الوقت هو بيبيع ياخد السوق بيبيع كويس جدا. دي نيفرز في مصر في مافيا رهيبه وفي سوق عملاق اكبر بكتير قوي من سوق تطعيمات البشر. It's a big field for the DNA vaccination.
هل احنا بنذكر الاستاذ الدكتور؟
نعم. اتس بروتين. انت بتاكل ايه يا حضرتك؟ لحمه مشويه صح؟ لا يعني اقول لك حاجه زي مثلا ما قالوا الجينيتيكال موديفايد، في ما زال في كونتروفيرشال في انجلترا وفي امريكا وفي كندا، لكن ولا ما ياكلوش. في ناس عارفه ياكلوه وفي ناس عارفه ما ياكلوش. ليه؟ مش مشكله. حضرتك متخيل كام ماده حضرتك بتقول لي في ماده وراثيه، متخيل كام ماده وراثيه تخش في معدتك كل يوم؟ حضرتك عندك بكتيريا في الامعاء اكثر من 15 نوع، كلهم بيحصل لهم ديجراديشن، الدنيا بتاعهم بيخش انترسيلر زي الدنيا فاكسين كل يوم. تمام؟ حاجه بتتنفس من الهواء، ده حاجه البكتيريا اللي لها علاقه اكتر من احنا نعرفها، وتخش كلها عندنا في الميوكس كل يوم، وكل يوم بيحصل لها تخش في البطن. احنا جسمنا بيشوف كل يوم آلاف الجينات الغريبه، وكل يوم جسمنا ديفلوب آلاف الجينات الغريبه. ومع ذلك سبحان الله ربنا خلق لنا في جسمنا سيستمز هائله بتعدي كل ده.
بنشكر الاستاذ دكتور ايهاب. في اي تعليقات ممكن في الانترفيو ان شاء الله؟
ان شاء الله ممكن بعد ما يخلص السيشن اي اهلا وزي ما قلنا مع الدكتور هاوس تسليم في الحاله.
شكرا يا
اي حاجه اهلا.
شكرا.